because she is the speakers. She is a member of the studies she has funded research project, and she has 2 design registrations filed. She has 25 publications in various journals of national and international repute, and she has been awarded the prestigious Professor J.G. Jolly Award in ISBTI 2022 and the AABB President's Award in 2022. Welcome, ma'am.
Thank you, Timas. It's always a pleasure joining the Timas academic sessions. So for today's meeting, I think we have got 2 very good speakers, Dr. Prateek Srivastava and Dr. Roshan. So it's a pleasure introducing both of them because I was with them at the ISBT Capetown meeting. So Dr. Prateek Srivastava, he's a registrar in the Department of Transfusion Medicine at the Medanta Hospital, Lucknow. He did his MD from PGI Chandigarh, and his achievements include the Harold Gunson Fellowship from the ISBT. He was also awarded a ICMR grant for his PG thesis, and he's currently working as senior resident in the Department of Transfusion Medicine at Medanta Hospital, Lucknow. So over to you, Dr. Prateek, for your presentation.
Starting with my presentation, which I presented in ISBT Capetown, and it was a study that I conducted during my MD thesis. It was on Buffy-coat pool platelets, a cost-effective alternative to single-donor apheresis platelets in hematology patients in Indian scenario, and it was a crossover trial. So I have the following disclosures. This study received the grant for from the Indian Council of Medical Research, and it was awarded with the Harold Gunson Fellowship by ISBT. So when we talk of platelet transfusion therapy, so there are a lot of conditions that require platelet transfusions, and the most common specialty which is a consumer of platelets are hematology disorders, hematology conditions like aplastic anemia, multiple myeloma, AML, ALL, and myelodysplastic syndrome. So initially, we were using 2 kinds of platelets in India. The first ones were the random donor platelets that have a volume of around 70 to 90 ml. The second ones were the apheresis platelets with a volume of 240 ml to 280 ml. Random donor platelets are byproducts of whole blood collection, whereas we need cell separators for apheresis platelets. So recently, there was an amendment in the Drugs and Cosmetic Act of 2020. And as per this amendment, there were a lot of changes that were inculcated related to blood banking itself. One such change was the incorporation of a new kind of platelet products, which was called the pooled platelets. So after this amendment, a third kind of platelet product was introduced in India, which is the Buffy-coat derived pooled platelets. So before we go further into the details of this study, I want to cover a little bit about what these individual platelets are about. So random donor platelets, they are the byproducts of whole blood collection. They have a lesser cost as compared to the other kind of platelets. They are readily available in our inventory, so we can issue them in the case of emergencies. And since the plasma content is very less, we can give them across the AVO barrier as well. The only problem with these platelets is that they have an insufficient platelet dose. So multiple units need to be given in order to attain the full platelet dose for a patient. The second platelets, which is the single donor apheresis platelets. Single donor apheresis platelets, they are the standard of care. These are the ideal platelet products. And the advantage of these platelets is that they have a they provide the patients with a full adult therapeutic dose, which is 3 into 10 to the power of 11 per bag. These are leukoreduced. The problem with these platelets are that they have a turnaround time of about 2 to 4 hours. So we cannot use them in the cases of emergencies. If there's an emergency and we don't have SDAP available with us. So in that case, the patient has to suffer. They have a lot of plasma, so we cannot give them across the ABO barrier. So they have to be transfused to ABO compatible recipients, and they come up with a considerable cost as well. So the third type of platelet, which was recently introduced by the Drugs and Cosmetic Act, which is the Buffy code pool platelets. So it has the advantages of both the platelets. For example, it gives a full therapeutic dose to the patient. These are leukoreduced because most of the hematoma patients who are undergoing platelet therapy, they require leukoreduced products for the prevention of HLA immunization, for the prevention of the viral borne infections and the other transfusion reactions as well. Third thing is that since these platelets are readily available in our inventory, prepare them. Depending on how we prepare them, if we suspend these platelets into platelet additive solution, so we can issue them across the ABO barrier as well. They have because the plasma is removed, so the chances of allergic reactions also go down, and it comes with a lesser cost because these are basically prepared from the byproducts of whole blood collection. So this is a study that was conducted in All India Institute of Medical Sciences, and this was conducted on dengue patients. And the authors, they found that the Buffy code pool platelets were fairly, similar in characters as compared to single donor apheresis platelets. However, these authors, they did not use platelet additive solution, so that part was missing. This is another study that was conducted at our institute at PGI Chandigarh. In this study, the authors, they have tried to calculate the basic biochemical parameters and the basic quality control parameters. They have compared and they have found that the platelets that are leukoreduced and PAS suspended have the best QC and the biochemical parameters. So coming to the aims and objectives of the study. So the first objective of this study was to find out if we are capable of producing Buffy code pool platelets on the very day of collection of the whole blood. So there are a lot of factors that we took into consideration because we had to calculate if it was feasible for us to calculate to produce Buffy code pool platelets on the very day of collection. The second objective was to check the efficacy of these products once they are transfused in the patients. The third objective of this study was to assess the safety of the product that we have produced. And finally, we also performed a cost analysis and compared it with that of SDAP. So coming to the methodology, this study was conducted at PGI Chandigarh between May 2021 till January 2022. We included only adult hematoma patients that were non-refractory to platelet transfusions. This was a randomized crossover trial and the sample size was 93. So how did we prepare pooled platelets? So basically, whatever whole blood collection we used to have, we used to sort out the O group whole blood, and once the Buffy codes that we prepared from these O group whole blood, we used to connect them from chain to chain method, and from the topmost bag we used to pass platelet additive solution, which used to rinse all these 5 bags, and in the bottommost bag the whole collected material was present. This bag we used the bottommost bag, which is the pooled Buffy code bag. We used to give them a soft spin. The details of the soft spin are mentioned in the inset on the left hand side, and we used to pass the supernatant through the inline filter, which caused the liquid reduction. And the final product was collected in the collection bag on the right hand side. So since we were pooling these platelets in platelet additive solution, and we were taking O group bags, so we wanted to see what the effect of suspension in platelet additive solution was bringing to the product. We wanted to see if the anti-A and anti-B titers were going down. So the internationally accepted critical high titer is that of 1:64. And in our study, what we found that the median anti-A titers of all the 93 bags that we produced was 1:8, whereas the median anti-B titer was 1:16. And the highest titer that we found amongst anti-A and anti-B was 1:32. So all the titers that we found were lesser than the internationally accepted criteria of 1:64 as the critical titer. So we only included the adult patients of hematoma that were non-refractory to platelet transfusions. we excluded pregnant females, we excluded patients who had undergone splenectomy, patients who were on drugs that can alter the platelet function like vancomycin, aspirin, and clopidogrel. So how did we do randomization? So basically, whenever we had a platelet requisition, whenever we used to have a platelet acquisition, we used to do randomization at the first step, and we used to select if this patient requires BCPP as the first product or SDAP as the first product. So if let's say the patient was selected as BCPP as the first product, so we used to transfuse this BCPP and then we used to check if the patient is refractory or not. If the patient was found to be refractory, this patient was excluded from the study. And upon the receipt of the second requisition, we used to give the alternate product, that is SDAP. Similarly, if the patient was selected first for SDAP, we used to perform CCI after 1 hour. If the patient was found to be non-refractory, only then we used to give the second product, which is BCPP in this patient. So this is the algorithm that shows the similar thing. It was mandatory for us to check if the product was safe for transfusion. So we conducted 1 hour post-transfusion CCI and PPR for all the patients. And we used to check for transfusion reactions as well. So coming to the evaluation of feasibility of BCPP production. So as I said, it was of prime importance for us to find out if we were capable of producing the bag on the very day of collection, because it is very challenging. There are a lot of factors. There are a lot of factors which are involved in the production of BCPP on the same day. For instance, we had to ensure that all the bags that were being collected in the camps had to reach the component separation lab by 1 p.m. Once we had all the bags, we used to do the blood grouping of all the bags, and we used to sort 6 units of whole blood that we would be using for the production of Buffy-Coat pooled platelets. We used to note down the numbers of all those 6 bags, and we used to send those bags for the CLIA testing. And the results of this TTI testing was made available to us before 6 o'clock of the same day. Only when all the 6 units were found to be non-reactive, we used to subject them for pooling. So coming to the evaluation of efficacy of the Buffy-Coat pooled products, we took into consideration 4 factors. The first one is the platelet increment after transfusion. The second one is corrected count increment. Third one is percent platelet recovery, and the fourth one is transfusion interval. So corrected count increment and percent platelet recovery, these take into account the body surface area as well as the total blood volume of the patient. So this is a comparative chart. As you can see that the QC characteristics of SDAP and BCPP were fairly similar, and based on these 4 parameters, we applied multivariate and univariate analysis, and it was found that based on the platelet increment and the transfusion interval, the Buffy-Coat pooled platelets were slightly superior as compared to the single donor apheresis platelets, whereas based on the corrected count increment and percent platelet recovery, they were non-inferior to single donor apheresis platelets. So producing a product that was safe for transfusion was of prime most importance in this study. So in order to ensure that we were issuing a safe product, we used to follow up the patients after every transfusion, and we used to do a medical examination to find out if the patient had any allergic reaction or febrile non-hemolytic transfusion reactions. For all the 93 patients that we transfused these products, no transfusion reactions were reported. Then on the 5th day of production, we used to send the samples for bacterial culture, and all the samples were found to be sterile. Since we were transfusing these platelets across the ABO barrier, so it was important for us to find out that once this product is being transfused into the patient, this patient was not developing any kind of hemolysis. So from the sample that we used to collect from this patient after 1 hour after transfusion, we used to run that on our HemeQ system to find out if there was any plasma hemoglobin that we could assess. However, we could not, there was no incidence of raised plasma hemoglobin, which indicates that no hemolysis was occurring in any of the patients. Coming to the cost analysis, because since we are working in a cost constrained setting like India, so it becomes very imperative to find out that the product that we are trying to introduce into our system has a lesser cost as compared to the product which is already there in the system, which is SDAP. So we took into consideration a lot of factors. For example, for human resources, we considered the salary of the technicians, of the doctors that are working in the blood bank, and how much time they are devoting for the production of BCPP and SDAP. For capillary infrastructures, we took out the cost of the camp, the component preparation room, the apheresis room, the camp cost, the fuel costs. For furniture, we took out the costs of the cold rooms, the blood bank refrigerators, air conditioners, agitators. Coming to the cost of medical equipments, there were the cost, we considered the cost of the bags, the SDAP kits, the tube sealers, etc. The cost of drugs, for example, we use calcium at times in the case of apheresis donors. We took into consideration the cost of consumables, for example, plated additive solution, indent reagent, gloves, and sanitizers. And finally, we also took into consideration the overhead cost, for example, the cold rooms, electricity, water, and biomedical waste. So as you can see, there are two tables. On the left-hand side, there is the annual cost of the product. So this is the bifurcation of the annual cost based on the various parameters that we have considered for each product. And we divided this annual cost by the total number of products that we were producing annually. So on the right-hand side table, you can see that the individual cost of the production of one SDAP was coming out to be 10,497 rupees, whereas the individual cost for the production of one bag of Buffycoat pool platelets is coming out to be just 4,063 rupees. So this is 61 times lesser as compared to the single donor apheresis platelets. So in conclusion, we can say that based on this study, in the case of non-refractory hematoma patients, Buffycoat pool platelets are non-inferior as compared to single donor apheresis platelets when it comes to 1 hour post-transfusion CCI and PPR, and superior in terms of platelet increment and inter-transfusion interval. Since Buffycoat pool platelets are costing very much less as compared to single donor apheresis platelets, so they can act as good alternative to single donor apheresis platelets in resource constrained settings like India. So after we conducted this study, we started issuing the Buffycoat pool platelets across the specialties, because for this study we were giving only to hematoma patients. After this study, we brought Buffycoat pool platelets into routine use, and now they are in clinical practice. So this study was not without limitations. There were a few limitations in the study. For example, the pre-transfusion titers of anti and anti-B for Buffycoat pool platelets could not be carried out. Since the patient load was very high at times, so we could not send the bacterial cultures of all the samples, only those platelets that remained with us in our inventory till the day 5. Only those specimens we could give the sample for. There are certain cases in which if the donor's platelet count is high, for example, if the donor's platelet count is 2.5 lakhs or above, we take double yield product for these patients. So that we could not take into consideration. And the impact of the donor's time was also not considered for the cost analysis of single donor apheresis platelets. So this is from my end. This is about the study that I conducted. Now, I'm sure there are a lot of fresh pass outs of transfusion medicine in our field who want to know about the Harel Gunson Fellowship. So I've included a few slides towards the end so as to orient them as to what this fellowship is about and how they can benefit from it. So basically, Harel Gunson is a fellowship which is offered by the International Society of Blood Transfusion. And it is a unique opportunity for fresh pass outs who have recently completed their thesis or those who have recently completed their MD degree. So ISBT holds conferences annually throughout the world, and it is attended by delegates from all over the world. So for every conference, the ISBT invites the abstracts for research studies from candidates who aim to present their study in these conferences. And these conferences, they have a lot of educational sessions. They have a lot of oral and poster presentations. There are workshops and exhibitions. So anybody who's starting out fresh in the career can actually go on to benefit a lot by this fellowship. So this is a chance for young doctors to attend the ISBT conferences. Around 8 to 10 such travel grants are awarded to candidates throughout the world. And the conference registration fees, the travel cost, the accommodation, and even the visa processing charges are covered by ISBT. And in order for visa application, the ISBT sends a visa application assistance invitation letter as well. So in order to apply for this fellowship, the age of the candidate should be 40 years or less. The applicant must be the first submitting and the presenting author of the accepted abstract. And in the case of high developmental index countries, only once you can avail this fellowship in your lifetime. But if you belong to a middle or low income countries, in that case, you can avail this fellowship twice in your lifetime before the age of 40. So the applications for Harald Gunson Fellowship, they are submitted at the time when you are submitting your abstract for the Congress. So once you've submitted your abstract, it is shortlisted by a panel of experts, and they shortlist the studies for oral or poster presentations. And out of all the selected candidates, only a few abstracts are selected who qualify for receiving travel grants. So Harald Gunson is more than just this is my experience, it is more than just a travel fellowship, because it is an acknowledgement of a good conducted research. And it provides an opportunity to especially to those candidates who are very in the earlier stage of their career, to attend an international conference. So this helps you gain insight into the global practices. So there are a lot of international audience. So this is an opportunity for you to present your work in front of an international audience and to receive critical feedback towards your study. So there's an opportunity to attend a lot of workshops which are free. And in ISBT Capetown, there were workshops on plasma fractionation. There were workshops on from SDFN, on e-learning. There were around 5 to 6 workshops which are free for the candidates. So you get an opportunity to attend this conference and to meet global delegates from your own profession who are like-minded. There are a few events like Young Professionals Breakfast and Harvard Gunson Awardees Lunch, which we got to attend. And this is a very rewarding and learning experience. So once you are back to your home country after attending this conference, you are required to submit a personal statement as to what you learned from during this conference. And this statement, it gets published in the next issue of Transfusion Today. And on the right side, you can see this is the ISBT website. If you want more information, you can always go into this portal and find out more information. So this is another initiative by ISBT. I'm sure most of the young, the freshers, they might not be knowing about it, but this is a very good initiative which is called the I Try It. Now, I Try It is an initiative by ISBT in which is a mentorship program. So they ask you for your abstract, and if you get selected, they mentor you regarding the intricacies of how to conduct a research. And this is an online as well as an offline. The lectures are online as well as offline. Offline, you get to attend these lectures in one of the ISBT congresses, and they teach you about the various components of research protocols and study design, and they help you to frame your manuscript and ultimately to get it published in a good journal. So this is a two-part program. The part one program it involves training in study design and protocol development, whereas part two is for those candidates who have done an initial part of the study, they have got the data, but they do not know how to implement that data, how to process that data, and how to write a manuscript based on that data. So they also give a small grant of around 5,000 euros for the successful candidates, and you get to attend the upcoming ISBT Congress. And the recordings of the previous year's ISBT is also available on the ISBT website. So this is this year's teaching roster of this iTrite program. And as you can see, there are a lot of topics which I've highlighted in the black box. For example, the practical data cleaning, developing tables and figures, writing tips, result selection, and all these are taught over a period of 5 to 6 months by 6 different mentors. So that is all from my side. If anybody wants to ask any questions, I'm happy to answer. And you can contact me on the email and WhatsApp number that I provided on the right-hand side in this presentation. Thank you.
Thank you, Prateek. I think it was a very good, your research topic was also very good. And whatever you have said about the I Try It and the Harald Gunson, that was also, I think, very useful. And I am sure there are I-Tried fellowship winners also here who can also share their experience. I think at the end of this webinar, they can also share their experience, how to go about. Thank you so much, Dr. Prateek.
Thank you, ma'am.
Yeah. So shall I invite the next person, Dr. Roshan? Can I have the Roshan CV, please?
Prateek, can you please stop your screen sharing? Thank you.
Yes. So thank you. The next speaker is Dr. Roshan Farida Fakruddin Shaikh. He is an assistant lab manager in the molecular genetics of Life Cell International Private Limited, Chennai, Tamil Nadu. He's again a recipient of the Harold Gunson Fellowship of ISBT Capetown 2023. Other awards include the Senior Research Fellowship CSIR, then Junior Research Fellowship of CSIR again, and 68th rank all over India. That is also very good. Then he is GATE qualified. His lectureship of the CSIR qualified. Late Letarau Scholarship, Best Outgoing Student. So many awards for Dr. Roshan. So I welcome Dr. Roshan. We are waiting to hear your presentation. Over to you, Dr. Roshan.
Thanks, ma'am. Let me just share my screen. Is the screen visible?
Yes, screen is visible.
Yeah, so good afternoon to one and all present here. So I'll be presenting a part of my PhD work, which basically involved identification of a novel mutation in Duffy antigen, which I presented in ISBT Cape Town conference. So it was basically a novel germline in-frame deletion in apparent Duffy B antigen individuals from Agri descendant, which was seen in India. So we know the fact that Duffy antigen was discovered in the early 1950s as a blood group antigen after they have identified the antibodies against the protein of a polytransfused hemophilic patient, and he was Duffy. Hence the name that for the antigen came from the patient. The study was then published in Nature in 1950 itself and authored by Mary, Mollis, and Dorothy. So it was basically an identification of a new human blood group. So what is basically a Duffy antigens? It's a glycosylated membrane protein, and the Duffy antigen serves as a receptor for several chemokines, including interleukin 8, GRO, RANTase, TARC, MCP1, etc. Surprisingly, it is also the receptor for human malarial parasites, mainly Plasmodium vivax and Plasmodium knowleski. It is structurally very similar to the G protein coupled receptors, which has 7 transmembrane domain. If you look at exclusively to the Duffy antigens, it consists of 338 amino acid, having an approximately 30 kilodalton weight. It is a very hydrophobic protein because of the 30% of its amino acids are hydrophobic in nature, and majority of the protein is buried inside the membrane. It's around 72%. if you look at the Duffy, it's basically encoded by the Duffy gene FY, which has two main alleles which are codominantly expressed, Duffy A and Duffy B. Now, when you say a codominantly expressed allele, basically indicates that both of the allele is going to produce a product and neither of them is going to be inhibited by each other. So if you have a parent which is Duffy A, the mother could be Duffy B, the children would receive a copy of Duffy A from father, a copy of Duffy B from mother, and the child would express both Duffy A and Duffy B on the red blood cells. So that basically, explains what is codominant in genetics. Since they are codominantly expressed, both the alleles, we have four phenotypes, which are seen in individuals majorly. So you have Duffy A, which basically indicates an absence of Duffy B antigen. You have Duffy AB, Duffy B, again an absence of Duffy A antigens on the red blood cells, and then you have Duffy nil. It's a total absence of either Duffy A and Duffy B on the red blood cells. When you look at the expressions of the Duffy antigens, these are not only expressed on the red blood cells, but they are indeed expressed on other blood cells, which includes endothelial cells lining of the blood vessels, epithelial cells of the kidney, lung alveoli, and Purkinje cells of the cerebellums. They are also expressed in the tissues of thyroid gland, colon, spleen, etc. Looking at the functionality of this protein, it's basically a glycoprotein and abbreviated in modern terminologies as DARC, which is Duffy antigen receptor for chemokines. The major function of this protein is to bind the chemicals that are secreted by the cells during inflammation and recruit other blood cells to the area of damage. It basically acts as the sink for the chemicals that are secreted during injury or inflammation or accident. Yes. So if you look at the genomic localization of the gene, it is present on chromosome 1 on the Q arm between 21 and 22 band, and the polymorphisms in the Duffy antigen is basically the basis for the development of the Duffy blood group systems. So how the transcription of the gene is regulated. So you have a Duffy gene, which is divided into two exons, exon 1 and exon 2. Exon 2 is the major one encoding for the entire Duffy antigens, and both the exons are intervened by a non-coding region called as intron 1. You have a promoter region which is called as the GATA, which is upstream of the exon 1. And here you have a very specific sequence which is recognized by the human GATA factor. Okay, so the major function of the human GATA factor is to recruit the RNA polymerase and other assembling enzymes to the region on chromosome 1 for the synthesis of mRNA followed by production of Duffy antigens. So if at the minus 33 region, if you have the nucleotide T, then the human GATA factor will come, recruit the RNA assemblies, and as a result of which mRNA will be produced, and you do get synthesis of Duffy antigen on both erythroid cells and non-erythroid cells. However, when you have a polymorphism, a polymorphism or a mutation at the very same position, now at minus 33 region of the Duffy gene, you have a transition from T to C. So, the genetic nucleotide, what we have ATCG, T has changed to C. Now, because of this, the human GATA factor is no longer able to recognize this sequence, and as a result of which, it is not able to recruit the RNA transcript assembly, and no RNA will be formed in the RBC, as a result of which the Duffy antigen will not be produced on the red blood cells. However, you do get production of Duffy antigen on non-erythroid cells. So you can let me know if you can know the answer why non-erythroid cells still has Duffy antigens expressed even you have change at -33 from T to C, or else otherwise I can let you know at the end of the presentation. So what we basically have is like genetic basis for Duffy antigen systems. We have majorly three polymorphisms which are seen in Duffy antigen. So the first and the major one is G125A, causing a change of the amino acid at position 42 G to D. So glycine is basically replaced by asparagine. So this is recognized by a restriction enzyme called as BAN1, and this is majorly the player to differentiate whether we have Duffy A or Duffy B. So using this enzyme BAN1, we can molecularly tell whether the person is Duffy A, Duffy B, or Duffy AB. Then rest of the two polymorphism, C256T, which basically arginine to cysteine, and G298A with alanine to threonine at 100th amino acid position, are the ones that are responsible for weaker expression of the Duffy antigen. The weaker expression could be associated with both Duffy A or Duffy B. However, in my studies, I have only encountered weaker expression of majorly of the Duffy B. So what basically happens, they reduces the stability of the antigen on the red blood cells, and you see weaker expression of the Duffy antigens on red blood cells on people who have these polymorphisms. So when you have when you do a serological testing and a molecular testing, and you have done your serological testing properly, identification of antigen using antisera on a fresh bloods, the blood has been collected recently, has been transported at proper temperature, and the antibodies used are like validated one, you will see a 100% concordance between the serological finding and the genetic molecular testing. So yes, so most of the time we do have problem with sample transportation, and we know with our experience that Duffy antigen, Duffy B especially, is quite an unstable molecules when it you store it for a longer time. Even in saline at 4 degree, it tends to get spontaneous shredding from the membrane. If you store like more than 3 or 4 days, you'll see definitely Duffy B transition from positive reaction to weak, very weak to negative reaction as well. So these are the places where molecular testing comes into picture. If you have the DNA, you can identify these polymorphisms and grade the person, whether the person has Duffy A, Duffy B, and whether these are like expressed at a weaker, whether the Duffy antigen will be expressed weaker or not, like that. So these are the major three polymorphisms that we also studied in our study. So what was the methodology that we have employed was, we have conducted a community blood collections, and subjected the collected blood for basically two testing involving serological testing for Duffy antigens and molecular testing, which involved extracting the DNA from the collected blood, performing a PCR followed by restriction fragment length polymorphism. And if we have seen anything aberrant on restriction fragment length polymorphism, we'll validate it on a Sanger sequencing. Now, whether we get or not get, you have to validate some of the data of RLP on Sanger sequencing. So, to our, I would say, I was lucky enough to find aberrant, novel finding on RFLP itself, and we have taken those data on Sanger sequencing. And in Sanger sequencing also, we figured it out that yes, there is something different and which has not been reported so far. So that analysis we have taken to online prediction tool by in silico means. So the results basically composed of both serological finding and molecular testing. So in serological testing, what we have found is that out of the 331 samples that were tested for Duffy, 326 samples showed a normal complement of Duffy A, Duffy B, or Duffy B. We did not encounter any Duffy null phenotype for you know mm, for among the 331 samples tested. However, out of 331, 5 samples showed weak to very weak agglutination under the microscopes, and even on gel card they were like, quite a weak strength, what we have figured it out. So these were taken for molecular testing, basically PCR, RFLP, and Sanger sequencing. So what we have found on molecular testing, so this basically RFLP, restriction fragment length polymorphism gel. So you have two panels, panel A and panel B. So what we have done is we have taken the PCR product and subjected the PCR product differently by two different restriction enzymes. First A by ACI1 and B by MWO1. So if you see the blue bracket basically shows the normal constituent from a normal individual. However, the green box indicates that there are certain extra band, upper part. So these were the one that triggered us to think over why these extra bands are seen over the gel, which are not seen in the normal complement in a normal individual. So the aberrant pattern were very much evident in both the gels, panel A and panel B. You see both apparent pattern same. What we have done is we took this PCR product, the undigested PCR product, for Sanger sequencing. So on a Sanger sequencing, we ran normal control like me, you, or could be anyone, could be normal, and the test sample. So the upper panel, panel A, basically indicates the chromatogram from a normal control and the test chromatogram. So um, the indicated arrow shows that, from this position there are peaks are mixed. So this basically indicates that there are something, either insertion, deletion, or something like that. So when we have analyzed the chromatograms, we found that there is a 3 base pair deletion between the coding sequence of 144 to 146, and because of this deletion of 3 nucleotide TGC, the amino acid at position 49 has been deleted from the um, protein sequence. So we know that it is a 338 amino acids. So when we compare both the wild type with the mutated Duffy antigen, we figured it out that this amino acid causes shortening of the protein by a single amino acids. Now, so the mutated antigen is counting to 337. We also have run our analysis in silico. So it's basically an online platform. So we tested our findings over two platforms. One is Proven, which is meant for protein stability, and the other one is mutation testing, that is meant for analyzing the genetic data. So when we subjected our finding of 3 base pair deletion involving a deletion of the amino acid in the protein, we found that Provin predicted it as the deleterious mutation with a score of minus 4.399. So usually the score of minus 2.5 is, considered to be normal or with no effect. Anything above beyond minus 2.5 should be considered as the deleterious one. And in our case, the Provin did predicted is to be deleterious one. And when we looked at the mutation testing again said that this polymorphism or mutation of 3 base TGC deletion in the Duffy antigen affects the protein features, and this was not reported in any of the databases that are available right now. So what we basically have done in our findings, so we have described a novel 3 base pair deletion, which is in frame deletion, which was identified again in 5 unrelated individuals of Agri descendant, and these were again found to be homozygous for Duffy B antigen itself. This novel deletion of alanine at position 49 of the transmembrane segment is seems to be fairly pathogenic by the two tested in silico platform, namely Proven and mutation tested, and we have seen in our serological finding that it complement the data as well. The presence of these mutations might have an implication for managing transfusion therapy this is an hypothesis. This we have not yet tested, and we don't know also. So it's just an hypothesis that we have given presumption. The mutation in exon 2 of Duffy gene significantly reduces its expression. This we know the fact because the serological finding did showed on the slide agglutination reaction as well. Sorry, not slide agglutination, tube agglutination reaction, and the gel card as well. And the in-frame deletion, we think, okay, this is again, it's our hypothesis, not yet tested, that it might not be directly related to the exposure of human genome to P-Vivax. However, it might have arisen due to de novo deletion mutation specific to Agri community. This the hypothesis is proposed basically because if it would have occurred because of the exposure of human genome to P-Vivax, this deletion should be fairly seen in other population as well. Agri community is such one, we know it, they are very close community, and inter caste marriage within that community is very high. Marrying a person out of their community, we see there is a very few incidents we have seen it. So probably this is the reason that we say that it's a de novo deletional mutation in the Agri community, and not because of the exposure of the human genome to P-Vivax. So what are the basically take home messages from our studies? That whenever we have a normal data, it becomes very easy for us to interpret it. Basically our life is easy. However, when we have an unexplained data or an abnormal findings, that could possibly lead to discoveries. And in our case, the discovery features that there is significance. If you use different investigation methodologies, it can help you to better comprehend your data. In our case, we could correlate the genetic basis of the blood group antigens in a much better understanding, provided with the serological testing as well. So that's complete my presentation. I would like to acknowledge everyone who were directly and indirectly connected to the studies, including my parents, guide, mentor, lab members, friends who were in and out of the lab, the participants, and ICMR for intramural funding. So if you have anything, I can take the questions.
So, Roshan, thank you very much for your presentation. It was something that we don't usually get to hear. You have discovered some new mutations in the Duffy antigen. So, after interacting with you, I would like to know, like, what is the work that you do in presently? Like, everyone may want to know, because it is not something that we are routinely exposed to. You're doing a different sort of work is being handled by you. So, please go ahead and tell us about the sort of work that you do in your current position.
Currently, I am occupying a position of assistant manager in LifeCell, and for the past, it's almost 2 years now that I'll be completing in July. That I'm heading the team of aneuploidy testing and Y chromosome microdeletion. So the work that I do, it's totally different that I did it in my PhD. Okay, transfusion medicine, it's not at all related to it. But currently what I'm looking is the genetic testing of the aneuploidies. So what we basically test is we look for chromosomal aneuploidy, which are very common. Okay, so you must have heard about trisomy 21, Edward syndrome, which is trisomy 18. Then you have Patau syndrome, which is trisomy 13, which are implicated in having a spontaneous abortion, excluding the trisomy 21. Trisomy 13 and 18 are very much responsible for spontaneous abortion. Apart from there, then you have trisomy 15, 16, 22, and Klinefelter Turner, and Jacob syndrome, which are the aneuploidy of sex chromosomes. Klinefelter is XXY, Jacob is XYY, and Turner is absence of one X from in the female. So female would be like only having or harboring a single chromosome. So my team, we work on such at the global level of the few chromosome, I would say, not the whole chromosome. We work on only 7 chromosomes, designated 7 chromosomes, and look after whether there is an aneuploidy in the prenatal sample. We do get very few samples, postnatal samples, but the fraction is less than 1%. So the current load is anywhere between 700 to 900 samples per month that we get it. So in that we see about 7 to 12% of the sample will have certain or the other chromosomal aneuploidy. So majorly we get an amniotic fluid for testing, which is around 90% of the total sample size that we get, followed by product of conception. These are uh, when we say product of conception, these are the product that are collected either from the spontaneous abortion, or when the baby is dead inside, it has been flushed out, they will send a part of the tissue to us for testing. And then the least that we receive is the Korean villus sample for testing. So after once the sample is received in the lab, within 36 hours, we will issue the report to the clinicians and the patient, telling them whether the child has abnormality for these designated chromosome or no. And if you are not able to tell them appropriately whether it is normal or abnormal, we will recommend them for other tests like chromosomal microarray or for karyotyping. That falls the very small fractions, like 2 to 3% of the samples that we get it. Apart from that, majority of the reports we will be releasing either normal or abnormal. And the other thing that I take care of is Y chromosome microdeletions. Again, it is an infertility panel that we look. So there is an region called as AZF region, AZF A, B, C, and BC regions. So this region on chromosome Y are responsible for proper production and maintenance of these sperms. Basically, we look after the infertility in the males. So if there is any deletion in these region, either of A, B, C, or BC, the male can have various disorders. So azoospermia or teratospermia, anthrosospermia, ethenospermia, like that. Azoospermia is total absence of sperm. Then you have teratospermia. The sperm's head would be crooked, or the tail would be crooked, like that, or the low count of the sperms. So that can be correlated if we do a Y chromosome microdeletion. So again, in Indian scenario, we find that yes, it's basically a filtered sample that we get in live cell. So that clinician refer us looking at the clinical history, and the couple are not able to have a child, after having an unprotected intercourse for more than a year, they're still not able to have a child. Then they will refer the samples to us to look at the Y chromosome microdeletion, provided the female would be like very normal, she doesn't have any problem. Obviously the arrow will be pointed toward the male partner then. So we will be testing, and to our knowledge, whatever the sample we get, around 40 to 57% of that has one or two major deletions in AZF region, which is directly correlated with impeding with the production of the sperm, or not improper maintenance of the cells, sperm. So there could be Sertoli syndrome, certainly only syndrome, or certainly in combination with other syndrome would be there. So yes, in past almost 1 year, 8 months, we have figured it out certain novel finding in Y chromosome microdeletion as well, like where we have received a sample from 34 years male. He was very much normal physically looking, photograph and all, we have seen it, have a normal descended scrotum, test is well developed, everything. So when we did a molecular testing with Y chromosome uh, Y chromosome microdeletion panel, we figured it out that he doesn't have a chromosome, Y chromosome at all. Means all the markers that I tested, like we test like 16 markers for Y chromosome microdeletion. So out of 16 markers, only 2 markers were present, rest were like not seen. So since I also have a QFPCR for aneuploidy testing, I immediately took that sample on aneuploidy testing and figured it out that this person is showing no signal for Y chromosomes. Then we recommended that person for karyotyping, and karyotyping then confirmed our finding as well that this person is an exo male, which is a very rare, kind of a finding that we have. The live birth rate or the finding incidence is around 1 in 20 to 1 in 25,000 in Indian scenario. So that is what we have figured it out. We told the clinician as well, and we are in a process of communicating this case study for publication as well. So yeah, these are the two major things that I look. Apart from that, there is like maternal cell contamination, which is exclusively done for prenatal samples. So we will test whether the DNA extracted has contribution from the mother, because certain molecular assays are get quite affected if there is a maternal cell contamination or the contribution of the DNA from the mother side. Certain assay will not tolerate even 1%, or certain will tolerate to certain percentage, and we can reliably release the report. My testing of aneuploidy can tolerate up to 20%, while like Sanger, next gen sequencing, or HLA typing, they don't tolerate even 1% of maternal cell contamination. So that also is taken by my team. So that's all, Amita ma'am.
Yes. That was very extensively you talked about, Roshan. Thank you so much. Just one more question. Like your
Hmm.
lab is accredited with any international agencies?
Yes, means we have for molecular testing, we are CAP accredited.
Okay.
Uh, yeah, so NABL accreditation has been done in this, not this, sorry. Yeah, this year, this only, March, not this March, sorry. Last March, yeah, last March we got a renewal for NABL accreditation, which is a 2 years renewal. So we will be undergoing again NABL accreditation next year. And CAP accreditation is done again twice in a year, and it is scheduled this year in anywhere between June or July. Apart from that, lifestyle has other accreditation as well, but those are like for non-molecular, like for because lifestyle is involved in stem cells storing and cord blood storing now. So they are
Mm.
accredited by AABB and ATBI, like that. So there are
That's
plenty
nice.
of other international accreditations that they have, and they are also there in production of certain pharmaceutical products. For again, they have their different set of accreditations.
Mhm. So that is another division under lifestyle.
Yes, yes, yeah, yeah. Though that is a totally different
Mm.
non-molecular. So we don't do that, NBS we called it as, and we come under NRL molecular. So that division is totally different, and we are not much aware of it,
Yes.
frankly speaking. We don't get time also, to
Yes.
this venture it out over there.
So I think anyone has questions, they can go ahead and ask it either in the chat box or by raising hands.
I just wanted to know whether you guys understood why I asked like if there is a mutation in the polymorphic region of the promoter region, the Duffy antigen will not be expressed on the red blood cells, but it will be expressed in other cell lines in the body. So, could anyone figure it out why? Otherwise, I can go ahead and tell.
So no responses have come up so far.
So it's a very simple answer to that. In red blood cells, the promoter region recognized by the human Gata factor is very specific to the promoter of the red blood cells. Okay, wherein in other tissues or cell line, the Gata region is not recognized by the human Gata factor. It is the some other factors of the promoter region that is recognized. So even though you'll have a mutation in the Gata region of the red blood cells, uh, your antigens of Duffy will be expressed in in the other lineage of the cell lines. Could be anywhere. So that is the reason why this promoter mutation is very specific to RBC. And even though that the very same mutation will be present in some other cell lines, but that will not be recognized by the RNA polymerase. So that is why, yes, even though you will not express the Duffy antigen on red blood cells, but you will see their expression in other cell lines.
Thank you, sir.
Dr. Prateek, are you there?
Yes, ma'am.
Yeah, just I wanted to clarify this. Apheresis platelet you are collecting in PAS?
No, no, ma'am.
So it
We
was
do
in
have an option in certain cell separators like amicus. We have
Ah.
an option of selecting between pass or actual plasma
Okay.
at the start of the procedure. But in our case, we did not collect SDAP in
Okay.
pass. We
Fine,
used
fine.
the
Okay.
patient's plasma itself.
You are having NAT testing there, or you are stopping with the CLIA?
Yes. No, we did have NAT testing for all the whole blood that we collected for Buffy code pool platelets. The NAT testing results were made available the next day.
Next day. So
So,
after CLIA, you
yeah.
started the polling. But the
Yeah,
NAT
yeah.
came later.
Pooling we used to do based on the CLIA testing, based on the serological testing, we used to go ahead with the pooling only, but we used to issue it only when we received the NAT results the next morning.
Okay.
So there was only one incident in which the serologically all the five pooled units were non-reactive, but one of them units were found to be reactive on NAT, so we discarded that unit the next morning. So
Yes.
otherwise, no, yeah,
Yeah,
it depends on the NAT
going
yield.
to
and all,
ask.
yeah.
Okay, okay. Okay, so there was only one case like that. Yeah.
Yes.
So you're continuing to go for the Buffy code, right?
Yes, ma'am. Yes.
Yes. So that was a very good study. You had looked at the logistics, you had looked at the feasibility, economics, everything you had looked. So that was a very excellent study. And thanks to both of you, Prateek and Dr. Roshan. Thank you, both of you. So I think if there are no more questions, we can call it a day.
Yes, ma'am. So thank you, Amita ma'am, for chairing the sessions today. And I also take this opportunity to thank the speakers for today, Dr. Prateek and Dr. Roshan. So the recorded session of today's TMAS webinar will be available in the YouTube channel of TMAS very soon. So that's all for today. Until we meet again for another session, goodbye.
Thank you, everyone, and thank you, Team Us.
Thanks everyone. Thanks Amita ma'am, and thanks Atira.
Thank you.